H unit. You're all part of the H unit now. You're my H unit, all my students and my peeps. And we're talking part 2. Oops, what did I just do? No, go back. Part 2, chapter 16, Holes anatomy, immune system. All right, let's start talking cell-mediated immunity. Can you actually hear me? Yes, you can. Okay, back to this idea of antigens and so on. So cell-mediated immunity is T cells. Okay, the T cells have to find out what exactly they need to attack, and this is why it's specific immunity here. Antigens are presented by macrophages to a T cell. So the macrophage, like Pac-Man, is going to eat the foreign invader. Let's see, I need a prop. Here's the foreign invader. Looks very vicious, right? Pathogen here eats this foreign invader and takes it all apart. It then takes pieces from that pathogen and it puts it on its own surface. Here's the macrophage in my hand and it puts it on the surface. It then travels over and is exposed to a helper T cell, and the helper T cell gets to see this antigen so that it can identify this specific antigen. And the helper T cell is kind of the conductor. Once the macrophage, which is the first on the scene, essentially identifies or shows this antigen, it's an antigen-presenting cell, then this helper T cell can then send out signals to all the others. These helper T cells help to recognize self from non-self, which is important. You don't want to send out an alarm against a cell that is presenting a self-antigen. And then after the antigen binds to this helper T cell, you get clonal selection of that helper T cell, just like we saw with B cells in the first part. So here's exactly what we're looking at. Here's the macrophage, consumes the pathogen, shows these little antigens on its surface, and then it exposes them to the helper T cell. This helper T cell is then going to communicate directly with both B cells and cytotoxic T cells. They'll do different things and attack in different ways, different types of cells, and so on. Cell-mediated is cytotoxic T. B cell is humoral immunity. Okay, so let's look at how we do these clones. Cytotoxic T cells, we used to call them killer T cells, but we don't want to confuse them with natural killer cells. So we call them cytotoxic T cells now, and they specialize in killing infected cells. Those things, for example, that a virus has entered and is reproducing inside the cell, it still looks to your immune system like a self cell because it is your own cell. It has to be targeted for destruction with these cytotoxic T cells. And cytotoxic T cells work kind of like, imagine a cell holding a syringe, and inside that syringe is a chemical called perforin, and they stab and inject the pathogen, one of your own cells with the virus in it. Think like the movie Psycho, where you have the stabbing team, and they're stabbing the cell with virus in it and injecting perforin. And perforin is going to create all these holes and degrade and disintegrate the cell so that it lyses. Helper T cells, which I already mentioned, recruits. It's the operator or the conductor, if you like, and they interact directly with B cells, much like they interacted with the cytotoxic T cells there. There's a last category which you probably haven't heard of much because a little bit less is known about them, but they're called suppressor T cells. This is like the negative feedback system of the immune system, so that while these other cells are going off and attacking, sometimes they can just get pathogen thirsty and try to attack everything in sight. These suppressor T cells help to keep them in check so that they don't go overboard. They release chemicals to suppress the activity of T and B cells. So someone with an autoimmune disorder could potentially have issues with their suppressor T cells where they're not down-regulating their production of T and B cells. They can't stop their immune response very well. And then, of course, all these different members, helper T cells, cytotoxic T cells, and suppressor T cells, all have their own memory cells. Should you encounter the same antigens again a second time, they can mount a response much more quickly and more severely. So this is a big summary of all this response, a great slide to know, where you have the first exposure of the antigen, macrophage eats it up and shows it to the helper T cell. The B cell also gets shown the antigen. The helper T cell will show the B cell as well and show the cytotoxic T. The B cell is going to mature into plasma cells and then secrete antibodies. And then the helper T cell is going to become a memory cell as well. And that memory cell can stimulate memory B cells, memory T cells, and so on, so that at second exposure, you can mount a response again second time. And these cytotoxic T cells will become active and defend against intracellular pathogens and cancer, and those cells that have been affected, like infected with viruses. That's what's meant by intracellular pathogens, pathogens inside the cells. This is going to defend against extracellular pathogens, these being bacteria. They are their own cell, as well as things like allergens and so on that we'll talk about here in a bit. Now, transplants and tissue rejection are an issue. Should you need them, there's different types of grafts or things to transfer. You can have an autograft, which is when one tissue is transplanted from one site to another on the same person. So, for example, if you need heart bypass surgery, they take a vein often from your leg or your arm and use that to supplant or to bypass, to go around the clot in one of your own arteries or veins of your heart. Isografts are between identical twins. Allografts is from an unrelated person, which is the one you typically hear from. And xenografts is from a different animal species, which is more unusual, but for example, sometimes pig hearts have been transplanted into humans. Very similar, same functionality. Those are all different kinds of transplants. Now, as you might imagine, autografts and isografts are the ideal because there's less chance of rejection, and xenografts are almost never successful, although sometimes they still try and for research purposes. And then allografts, you have to go through a whole series of steps to identify and confirm that the set matches, that the organ matches the recipient. Okay, allergies. Many of you have allergies, which are basically hypersensitivities to certain environmental things that you shouldn't be hypersensitive to. It's an abnormal immune response, and there's different types. There's immediate hypersensitivity, where right away you're exposed to the allergen and you form a response. And this comes from the release of histamine, where IgE binds to mast cells. So in your body you have a cell type called mast cells, and the immunoglobulin, if it's floating around in your bloodstream, IgE, if it binds to mast cells, those mast cells in that area are going to release histamine. And histamine triggers an an inflammatory response, where you get red and swollen and itchy and that type of thing. And this happens within seconds of contacts to the allergen. Under severe circumstances, you can go into anaphylactic shock, such as like a bee sting. People will have an EpiPen I did this as if it's a pen, an EpiPen, where if they get exposed to the allergen, like stung by a bee, they have to inject epinephrine to block their immune system's function, to block the inflammatory process. In most people, this is not the case. Most just have simple allergies like mine to cats. If I'm at a friend's house with cats, I start to get red and itchy, and I hate it. I wonder why we dissect cats. Sorry, off on a tangent. Types of allergies. That was immediate hypersensitivity. You can also have delayed hypersensitivity, which is triggered from the release of lymphokines, a type of chemical that comes from helper T cells. This is not the immediate type of thing where you think of allergens like bees or pollen or whatever. This usually happens 1 to 3 days after contact with the antigen. And the problem is that we don't really know what the antigen was, because it's so far after the exposure that you get the response that you don't know exactly what caused it. It's often difficult to determine with delayed hypersensitivity. Now, here is showing you exactly, or essentially, how it works. Go back. Here we have IgE binding to, it's released from a plasma cell, because IgE is an antibody, and it works against allergens. IgE antibodies are going to attach to mast cells in certain body tissues, and then this is going to trigger, when this antigen binds to this IgE, if it fits, so the variable region of the antibody of IgE, if it binds to this antigen that it's been primed for earlier, then it's going to release all this histamine. And this histamine, again, triggers inflammation. It triggers blood vessel dilation, capillaries become leaky, it stimulates release of large amounts of mucus, causes smooth muscles to contract, and so on. So lots of fluid leaves and exits into that region. It's called inflammation or swelling, if you like. Okay? Now, there are certain disorders of immunity, of course, immunodeficiencies, which can be congenital or acquired, meaning you can be born with them or you can acquire them. One of the acquired ones would be AIDS, acquired immune deficiency syndrome. It's triggered or caused by the HIV virus. And AIDS is characterized by having a very low white blood cell count because HIV infects the helper T cells, and when HIV starts to pop out and kill off those helper T cells, then you lose the ability to counteract normal, typical types of pathogens like the flu or the cold, and you typically will, if you die with AIDS, you die of exposure to the flu or cold, that type of thing. More commonly, you hear of things that are disorders of the immune system, specifically autoimmune diseases, where the immune system cannot properly distinguish self from non-self. And in this case, the body is going to produce antibodies and cytotoxic T cells that are going to attack itself. Okay? Examples include MS, where the white matter of the brain, specifically the myelin of the brain, is targeted and killed, so they can't properly send messages at the proper speeds anymore. Myasthenia gravis, we've learned about before, communication between nerves and skeletal muscles, specifically the neuromuscular junction. Type 1 diabetes is when the pancreatic beta cells, which produce the insulin, get attacked by the immune system and they slowly disappear. You don't have those cells to produce insulin anymore. Rheumatoid arthritis is one of the two types of arthritis. This is not the one from overuse. This is an autoimmune disorder. There is arthritis which just from using your fingers or whatever too much, they slowly start to build up scar tissue. But in this case, rheumatoid arthritis, the immune system attacks the collagen and the connective tissue inside the joints of commonly used areas, fingers, toes, that kind of thing. That is autoimmune. Lupus, systemic lupus, people just call it lupus, affects all different kinds of things. That's autoimmune. And glomerulonephritis, try and say that 5 times fast, basically is attacking your own kidney. Okay. Now, how does the lymphatic system and body's defenses develop? We've talked about this before. We'll go through it again really quickly here. Thymus and spleen are the only ones there before birth. Okay, all the other ones are very poorly developed. Your immune system basically sucks before you're born. Okay, a newborn actually has no functioning lymphocytes at birth. You don't have B cells, you don't have T cells. If you get sick in the first 3 months as a baby, you're kind of screwed because it's very difficult to defend. If you are breastfed, it's a lot easier because mom is giving you her antibodies through the breast milk, IgA, and that's what we describe as passive immunity, right? You might remember that. And this is why, for example, basically, new mothers keep their babies indoors and they don't let a lot of people, or they shouldn't let a lot of people come and see the baby and interact with it and so on because they don't want to get it sick. Sometimes you might need to remove lymphatics or specifically lymphoid tissues, and edema can result because the tissues are a place where the lymph is flowing through, and if they're not there, you can get a buildup of the lymph at the region where they're supposed to continue passing through, and we call this edema, a buildup of fluid. Over time, these vessels will grow back and continue because they do continue, they do finish and grow. And on that note, we are done with Chapter 16. I hope you learned something today because I sure did.